It really was an extremely exciting finding. It made total sense that the connections would be there, but we knew immediately it's going to take several years to convince the field that is the case, because it is so unheard of, or it was so unheard of that cerebellum could be doing something non-motor and so powerfully. We have always known that there is a link between cerebellum and addictive behavior, but we could never understand mechanistically how this could come about. This is the first time that we have managed to demonstrate a direct connection between cerebellum, part of the brain that has historically been thought to control motor coordination, with another part of the brain that is involved in non-motor functions, ventral tegmental area in this case, which is involved in reward processing. The finding that cerebellum directly projects to the ventral tegmental area is quite important. I think neuroscience textbooks, neurology textbooks may have to be modified to indicate this, and will have fairly profound impact on our understanding of cerebellar function. Perhaps what cerebellum is best known for is control of motor function and movement. We realized that there is evidence in humans that cerebellum might also be playing non-motor functions. In patients, there is very strong association between changes in the cerebellum in terms of its activation or morphology and psychiatric disorders such as schizophrenia and autism spectrum disorder. We suspected that there might be a connection between the cerebellum and the VTA, ventral tegmental area, one of the major brain structures that provides dopamine to the rest of the brain. This is really quite important because dopamine is a reward signal to propel you to do motivational behavior for things that are important for your survival. This work was done by two very talented students in my lab, Chris Chen and Ilaria Carta, together with Shanad Doriz and Amanda Schott. We performed a number of anatomical studies, and indeed we could see that there were projections from cerebellum to the VTA. We wanted to know whether these projections were functional, and if so, how powerful they were. We did a series of behavioral experiments in which we allowed the mice to explore an open field. On the left-hand side, you can see this open field and the mouse itself running around. And on the right-hand side, we have a heat map where the amount of time that the mouse explores and spends at each location is plotted in a color plot. You will find that the mouse explores all four quadrants to the same extent. Now what we can do is to randomly assign one of the quadrants to be the reward quadrant. So the moment that the mouse enters that quadrant, the computer recognizes this and delivers a series of light pulses to optogenetically activate the cerebellar input to the ventral tegmental area. If it is rewarding, if activation of these axons to the VTA make the animals feel good, they will keep coming back to that quadrant. And in fact, the mice rapidly learned a strategy of leaving and coming back to the reward quadrant so that they can have this feeling of pleasure. This experiment supported the hypothesis that the cerebellar input to the VTA provides reward. Next, to explore whether the cerebellar VTA projections can produce addictive behavior, we did a task in which you divide a chamber to two different parts, where one side was dark, the other side was light. Mice prefer to be in the dark side, but now what we do is to confine them to the bright side, but at the same time activate the cerebellar inputs to the VTA, and then we put the mice back, allow them to explore both chambers. And the expectation would be that they will go to the light side because they had the cerebellar activation of the VTA, and indeed that is what we found. So these experiments suggest that cerebellum can be involved in addictive behaviors. While our studies are quite at the earliest stages and are providing the foundations for understanding how the cerebellum plays a role in addictive behavior, in principle, if we could tease out the circuitry a little bit more, and for example, if we were to find that cerebellum is required for relapse, one could imagine manipulation of this pathway either pharmacologically or optogenetically with deep brain stimulation or transmagnetic stimulation to prevent relapse or to help patients come off drugs of abuse. We were very much interested to know whether the cerebellum could be playing a role, particularly in autism, in terms of social behavior. So what we did was to inactivate or inhibit the projection from cerebellum to the VTA. What we found was that mice could not socialize anymore. Autism spectrum disorder is a very complicated disorder, and the more we understand about the different brain circuitry that are involved in control of social behavior, the more we are in a position to provide therapeutics and help the patients. In the old days, one used to be able to say, this region does this, and then this region sends the information to region C, and then D and F. Now we understand that is not the case anymore. It seems that the brain processes information in parallel. So lots of regions simultaneously are doing things and are interacting with each other, and it's much more difficult to say who did what first. And that is perhaps why our brain is about 100 times faster than the fastest supercomputer we have ever made. And that's just amazing and beautiful. There's nothing more beautiful and exciting than the brain.